Hello, my name is Isabella Francisco. I have researched with Dr. Chai in the Department of Chemistry at John Carroll University on this project entitled Role of S-glutathionylation on Chicken Breast Extract. The main goals of this experiment were to analyze chicken breast fractions for protein components that were modified with glutathione disulfide GSSG using the technique of affinity chromatography. The proteins were modified with different concentrations of GSSG, and the presence of modified proteins was observed through the use of SDS page and Western blotting. First, it is important to define what S-glutathionylation is. It is a form of reversible post-translational modification in which a protein with a cysteine residue forms a disulfide bond with reduced glutathione. Glutathione is a very important molecule because it is the most abundant non-protein antioxidant and has a cysteine residue that is of the utmost importance in S-glutathionylation. S-glutathionylation is very important to study because of its role as a mechanism to protect and regulate many different situations in the cell, including protecting proteins in the cell from oxidative damage, which can lead to destruction and disease. The crude chicken breast extract was analyzed in this experiment because the fractions are very rich in protein. Since they were rich in protein, it was easy to observe the modification of these proteins. Additionally, the major protein that we were trying to identify was glyceraldehyde 3-phosphate dehydrogenase, also known as GAPDH. This is an enzyme that is important in the glycolysis pathway. It produces a very high energy intermediate called 1,3-bisphosphoglycerate, which can provide energy in the form of ATP for the cells, as well as NADH. The chicken breast extract fractions were separated using affinity chromatography. Fraction 1 is the crude chicken breast sample. Fraction 2 is the flow-through, or the components of the chicken breast extract that were not attracted to the reactive blue agarose beads in the column. These beads had cibricon blue attached to them that made them attractive to certain types of molecules. Lastly, fraction 3 includes the components of the chicken breast extract that were attracted to the column and eluted off with buffer and NADH. These different fractions were then incubated with different concentrations of GSSG. All of the fractions were first incubated with DTT, which is a reducing agent, for 30 minutes at 30 degrees. The use of DTT ensures that the protein cysteine residues are reduced so that they can undergo S-glutathionylation with glutathione. Different concentrations of GSSG, including 10, 25, and 50 millimolars of GSSG, were incubated with the different samples for 30 minutes at 30 degrees. A time-dependent reaction was also conducted with fraction 2. The fraction was still incubated with DTT for 30 minutes at 30 degrees with 2.5 millimolar and 5 millimolar GSSG, but the incubation times varied between 10, 20, and 30 minutes for each concentration of GSSG. After these samples were prepared, they were analyzed using SDS page and Western blotting. SDS page is a technique used to separate a protein of interest based on molecular weight. After this, the proteins were transferred from the agarose gel to a membrane through a process called Western blotting. After the transfer, the protein was blocked with milk solution to prevent antibody staining in areas other than the protein of interest. The membrane was then incubated with primary and secondary antibody. For the concentration and time-dependent reactions of fraction 1, 2, and 3, the primary antibody was anti-glutathione and the secondary was anti-mouse. For the last experiment, where we were identifying the major protein in fraction 3, the primary antibody was anti-GAPDH antibody and the secondary antibody was anti-rabbit. The results showed significant protein modifications in the different fractions. There were significant protein bands modified with GSSG at 33 kilodaltons in fraction 1, as shown in figure 1. In fraction 2, there were significant protein modifications, 16, 36, and 44 kilodaltons in fraction 2. And in fraction 3, at 36 kilodaltons, there was a major protein modification in fraction 3. All three of these experiments in figure 1, 2, and 3 analyzed how changing the concentration of GSSG modified the extent to which the proteins were modified. Additionally, it was observed that as the intensity of the band increased, the concentration of GSSG also increased, which means that a greater concentration of GSSG will result in a greater number of modified proteins. The next experiment was a time-dependent analysis of GSSG modification. These experimental results were included in figure 4. There were two significant modified bands at 176 and 65 kilodaltons. Lastly, the major protein in fraction 3 was identified to be glyceraldehyde 3-phosphate dehydrogenase, which was determined by using the primary antibody anti-GAPDH. This identification of GAPDH using anti-GAPDH as the primary antibody is included in figure 5. From these results, the identity of these modified proteins can be suggested. GAPDH has a molecular weight of 36 kilodaltons, and as seen in figure 5, the major protein was observed at about 36 kilodaltons using anti-GAPDH antibody. Therefore, this major protein band can be suggested to be GAPDH. It can also be strongly suggested that the protein bands at 44 kilodaltons in figure 2 is actin, which has a molecular weight of 42 kilodaltons. It can also be suggested that the band at 16 kilodaltons in figure 2 is a ubiquitin carrier protein. This ubiquitin carrier protein has a cysteine residue that can be modified with glutathione. Therefore, the goal of analyzing chicken breast fraction through the technique of affinity chromatography for protein modifications by GSSG was fulfilled throughout the experiment, and some of the protein bands, most notable GAPTH, were identified. I would like to thank the Barrett Endowment Calder and Weaver Undergraduate Research Fellowship, as well as Dr. Chai. Thank you.